students, today we will gain knowledge about microscopic examination of food, culture techniques, foodborne infections and intoxicants. And our main objectives will be to understand various techniques of microscopy methods that are used for the analysis of food, to understand the properties of basic cultural techniques in food microbiology, to gain knowledge of various foodborne diseases and intoxicants, to ascertain the factors that are responsible for foodborne infections. Friends, let me first introduce you the term food. Food is defined as a substance taken or absorbed in the body of an organism in order to sustain growth or repair, support vital processes and furnish energy for all activities of the organism. It is edible substance, usually of animal or plant origin, consisting of nourishing and nutritive components such as carbohydrates, fats, proteins, essential mineral and vitamins, which when ingested and assimilated through digestion sustains life, generates energy and provides growth, maintenance and health of the body. Now friends, let's learn about the food microbiology. The examination of food for the presence, types and numbers of microorganisms and their products is basic to food microbiology. In spite of the importance of this, none of the methods in common use permits the determination of exact numbers of microorganisms in a food product. Although some methods of analysis are better than others, every method has certain inherent limitations associated with its use. Microscopic examination or microscopy represents a cheaper alternative for the examination of food, and its use in food processing dates back to 1850 when Arthur Hassel used a microscope to tell coffee from chicory. His results were elaborated by other authors in the ensuing years. These researchers endeavored to combine the methods of microscopy with the most modern methods from other disciplines as well, such as immunohistochemistry, image analysis and serology. The work of the food microscopist encompasses a wide range of different activities. These range from a fundamental understanding of the food structure and function of raw materials and final products through studies of machinery and equipment that are used to manufacture food and the various materials that are being used to package food, to troubleshooting problems such as foreign bodies, faults in the food itself or poorly performing food packaging materials. In the last few decades, the full potential of electron microscopy has been recognized due to the fact that the information is rendered in a pictorial form. However, if needed, the microscopic image can be converted into numerical data that lend themselves to statistical processing. Coupled with variety of investigative techniques, today, there are many ways of processing and preparing samples for microscopic examinations, such as three-dimensional imaging, minimum sample intervention, and in situ microscopy that are used for dynamic studies to derive quantitative information from microscopic images. Modern food microscopy can be studied to determine components of food and its quality, recognition of food adulteration and harmful ingredients in food, and quantification of food components. Among the various imaging techniques, most widely used are light microscopy for studying the microstructure, while as transmission electron microscopy as well as scanning electron microscopy possessing higher magnifying imaging powers are used to study the ultrastructure of foodstuff. Now students, let's understand each of these techniques very briefly. Light microscopy that exists with a variety of modifications such as microscopy with transmitted light in the visible spectrum that works with samples prepared by specific or basic staining to differentiate the individual internal structure. The common staining is basically used to enhance all structures present in the final product, which are then identified by their morphological characteristics such as shape, size and the mutual cell configuration, the presence of crystals, grains, etc. However, special staining endures highlighting the selected structure with a different color than other parts of the product. To determine collagen, the main component of connective tissue, staining with green or blue trichrome or by means of picrocereus red is quite suitable. Similarly, alizarin red can be used for detecting the presence of bone fragments, and Sudan third, oil red or Sudan black to highlight the presence and structure of adipose tissues. Using these staining procedures, it is possible to analyze not only the dispersion of fat in the product but also its formation in the sub-packing layers. Friends, another microscopic technique is TEM, that is transmission electron microscopy. Transmission electron microscopy is electron optical instrument that are analogous to light microscopes. However, the specimen is illuminated by an electron beam as gas molecules would deflect the electrons. This requires operation in a vacuum. An electron gun at the top of the microscope column and a system of electromagnetic lenses focuses the electron beam on the sample. The interaction of electrons with the material, that is electron scattering, results in the image contrast in TEM. The image resolution is directly proportional to the acceleration voltage of the electrons. High resolution is obtained due to the short wavelength of the electrons when the voltage increases. However, increasing acceleration voltage leads to poorer contrast since scattering of electrons is decreased in the higher velocity. In TEM investigations of colloidal systems, voltages between 80 and 200 kilovolts are usually employed. However, we have another technique that's called cryo-TEM that allows for the direct investigation of colloids in the vitrified frozen hydrated state, and information about the internal structure of colloidal system is obtained. By preparing complex sample, the formulation microstructure is displayed in its original state and a clear differentiation between nano-sized droplets and other structures can be obtained. Now friends, let us discuss another microscopy technique, that is SEM, scanning electron microscopy. In scanning electron microscopy, the image is formed step by step by examining a focused electron beam across the specimen. The primary electrons penetrate the solid specimen and are deflected by a large number of elastic scattering processes. The incident electrons results in the generation of various signals which are collected to form an image or to analyze the sample surface. These are mainly secondary electrons or in other applications backscattered electrons with energies of a few tens of electron volts from the primary beam and the characteristic X-rays and are used to form an enlarged image of the specimen surface. The images are observed on a monitor and photographed using a 35 mm camera from a special monitor. Some advantages of SEM include relatively simple sample preparation, wide range of magnification, high depth of field, and the fact that the image is a representation of electronic data, allowing for image analysis and quantification. Direct method of observing the ice crystals of frozen samples without prior thawing is done by cryo-scanning electron microscopy. Cryo-scanning electron microscopy involves additional step by coating with metal for enhanced conductance of the electrons. The frozen specimens prepared for cryo-electron microscopy are not replicated but are immediately transferred to a low temperature stage within a microscope and viewed directly. This technique provides rapid physical fixation and avoids the risk of introducing artifacts entailed in chemical fixation. Structure collapse or shrinkage as sometimes occurs in typical preparation methods. Therefore, the advantages of cryosem include the preservation of triphasic structure of a sample with the distribution of solid, liquid and gas in a state closer to its natural state. However, its high cost is its main drawback. Another technique is environmental SEM, which differs from conventional SEM mainly by the presence of a gas in the specimen chamber. Samples are thus not viewed under high vacuum but under a deteriorated or low vacuum. This is possible because of a special design of electron optics column that allows differential pumping. The column is divided into different pressure zones separated by pressure limiting apertures. Another versatile and powerful microscopy technology used for studying samples at nanoscale is atomic force microscopes. The atomic force microscope is one kind of scanning probe microscopes. Scanning probe microscopes are designed to measure local properties such as height, friction, magnetism with a probe. An atomic force microscope uses a cantilever with a very sharp tip to scan over a sample surface. As the tip approaches the surface, the close range attractive force between the surface and the tip causes the cantilever to deflect towards the surface. However, as the cantilever is brought even closer to the surface such that the tip makes contact with it, increasingly repulsive force takes over and causes the cantilever to deflect away from the surface. A laser beam is used to detect cantilever deflections towards or away from the surface. By reflecting an incident beam off the flat top of the cantilever, any cantilever deflection will cause slight change in the direction of the reflected beam. A position sensitive photodiode can be used to track these changes. Thus, if an atomic force microscope tip passes over a raised surface feature, the resulting cantilever deflection and the subsequent changes in the direction of reflected beam is recorded by the position sensitive photodiode. Now friends, let us discuss some common cultural techniques that are used in food microbiology. In food microbiology, the examination of foods for the presence, types and number of microbes and their products is essential. However, as I already said, none of the single methods allows the determination of exact numbers of microorganisms in a food product because every method has certain inherent limitations associated with its use. Here, we will discuss some common cultural methods that are employed. First, standard plate count for viable cells. By the conventional standard plate count method, portions of the food samples are blended or homogenized, serially diluted in an appropriate diluent, plated in or onto a suitable agar medium. The properties of agar make it so useful to form a gel at low concentrations, which does not significantly influence the water potential of the medium. The media is then incubated at an appropriate temperature for a given time, after which all visible colonies are counted by use of a cubic or electronic counter. The SPC is by far the most widely used method for determining the number of viable cells or colony forming units in a food product. However, SPC procedure for selected groups are further limited by the degree of inhibition and effectiveness of the selective and differential agents employed. Although the SPC is more often determined by poor plating, essential comparing results can be obtained by surface plating. By surface plating method, pre-poured and hardened agar plates with dry surfaces are employed. The diluted specimens are planted onto the surface of the replicate plates, and with the aid of bent glass rods, the 0.1 ml inoculum per plate is carefully and evenly distributed over the entire surface. Second cultural method is most probable number counts. An alternative method of enumerating low number of viable microorganisms is by most probable number method. In this method, dilution of food samples are prepared as for the SPC. 3 serial aliquots or dilutions of the material to be studied are then planted into 9 or 15 tubes of appropriate medium for the 3 or 5 tube method respectively. The medium used has to be designed to make it possible to decide whether growth or no growth has occurred, and the number of positives at each sample size or dilution is determined after incubating the tubes. The method is statistical in nature and MPN results are generally higher than SPC results. This method was introduced by McCarty in 1915. It's not a precise method of analysis. The 95% confidence intervals for a 3-tube test range from 21 to 395. Although Woodward concluded that many MPN values are improbable, this method of analysis has gained popularity. There are computer programs for generating MPN values from different designs of the experiment, and these programs can also provide confidence limits for the MPN and suggest that the likelihood of particular combinations of positive results should be. A modern variation on the MPN theme is the use of hydrophobic grid membrane filter. A sample is filtered through the HGMF, which is divided by a hydrophobic grid into a number of small cells or growth compartments. After incubation of the filter on an appropriate medium, each of these cells is scored for growth or no growth. Third type is dye reduction. Dye reduction techniques are used to estimate number of viable cells that possess reducing capacities. 2 dyes commonly employed in this procedure to estimate the number of viable organisms in suitable products are methylene blue and rizazurin. To conduct a dye reduction test, properly prepared supernatant of foods are added to standard solutions of either dye for reduction from blue to white for methylene blue and from slate blue to pink or white for rizazurin. The time for dye reduction to occur is inversely proportional to the number of organisms in the sample. Methylene blue and rizazurin reduction by Hunden cultures were studied in milk. These tests, which have been used for some time in the dairy industry, depend on the response of a number of redox dyes to the presence of metabolically active microorganisms. They are relatively simple and rapid to carry out at low cost. The redox dyes are able to take up electrons from an active biological system, and this results in a change of color. Usually, the oxidized form is colored and the reduced form colorless, but the triphenyl tetrazolium salts are an important exception. Direct microscopic count. It's another technique. DMC is used for both viable and non-viable cells. In its simplest form, the DMC consists of making smears of food specimens or culture onto a microscope slide, staining with an appropriate dye and viewing and counting cells with the aid of a microscope oil immersion objective. DMCs are most widely used in the dairy industry for assessing the microbial quality of raw milk and other dairy products, and the specific method employed is that originally developed by R.S. Breed. Briefly, the method consists of adding 0.01 ml of sample to a 1 square centimeter area on a microscope slide and following fixing, defatting of sample and staining the organism or clumps of organisms are enumerated. The method lends itself to the rapid microbiological examination or other food products such as dried and frozen foods. Now friends, we will study some foodborne infections. A foodborne infection is a disease caused by ingestion of food contaminated by an agent, chemical or biological. Food poisoning is acute enteritis characterized by diarrhea, vomiting, with or without fever and abdominal pains. Food poisoning is normally associated with the small and large intestine. Certain types of food poisoning are described as intoxications and others as infections. Foodborne diseases range from relatively mild, self-limiting gastrointestinal upsets through to life-threatening conditions such as botulism. Some foodborne infections can develop severe complications such as hemolytic uremic syndrome associated with about 10% of E. coli infections, or the neurological disorder Guillain-Barré syndrome that follows about 0.1% of Campylobacter infections. Some groups of the people are particularly susceptible to the more serious consequences of foodborne disease. Intoxication involves food poisoning, in which the organism grows in food and releases a toxin from the cells. When the toxin is ingested along with the food, it gives rise to the food poisoning syndrome. Illness may occur as a consequence of ingestion of a preformed bacterial or mold toxin due to its growth in a food. A toxin has to be present in the contaminated food. Once the microorganisms have grown and produce a toxin in a food, there is no need of viable cells during the consumption of the food for illness to occur. Bacterial toxins that produce intoxications are the exotoxin types of either enterotoxin that are affecting the gut, as in the staphylococcal intoxication, or neurotoxin affecting the nervous system, as in botulism. Another category of intoxications are the mycotoxicosis due to the ingestion of mycotoxins and the disease caused by algal toxins, shellfish poisoning. Generally, intoxications have a short incubation periods. Food poisoning. Food poisoning is also caused by the ingestion of live organisms. The organisms grow in the gastrointestinal tract to produce the disease. Most microbial food poisoning fall in this category. For example, salmonellosis caused by Salmonella species like Salmonella typhi. Salmonella causes a gastroflu-like infection which in most cases lasts about 2 to 5 days. However, in some people, they can lead to chronic conditions such as Retter's syndrome or reactive arthritis. Salmonella usually needs to grow to reach high enough numbers to make healthy adults sick. Enteritis associated with food poisoning infections is due to the production of exotoxins or endotoxins that act as enterotoxins. In certain other types of food poisoning, as in the case of Clostridium perfringens, live cells need to be ingested for the disease to occur, but the organism does not grow and reproduce in the gut. Vegetative cells sporulate after ingestion and enterotoxin released, causing the disease symptoms. Similarly, food poisoning by Campylobacter is probably one of the most common causes of foodborne illness. Campylobacter is present in the gut of a wide range of animals, especially birds. Outbreaks have been linked to the consumption of undercooked poultry, mince and sausage, unpasteurized milk and cooked foods that have been contaminated by raw foods like meat and poultry. Listeriosis is a comparatively rare form of foodborne illness that's caused by Listeria monocytogenes, but it can be a very serious disease in a small group of individuals. It has also caused occasional outbreaks of mild gastroenteritis in healthy people. The symptoms are usually described as flu-like, although vomiting and discolored urine also occur. Staphylococcus aureus, also known as golden staph, as its important form both a medical and food perspective. Staphylococcus produces a heat-stable toxin as it grows, and it is the toxin that makes us sick. If it is allowed to grow in a food, the toxin will remain even if the food is cooked again. The toxin takes only a very short time to make sick, 1 to 6 hours, and causes nausea, vomiting, abdominal cramps and diarrhea as the usual symptoms. Since 1975, a number of Bacillus species have been associated with foodborne illness. For instance, Bacillus cereus causes 2 different types of food poisoning. the diarrheal type and the emetic or vomiting type. The diarrheal type of food poisoning is caused by an enterotoxin produced during vegetative growth of Bacillus cereus in the small intestine, while emetic toxin is produced by cells growing in the food. Foodborne infections vary in their mode of action on the gastrointestinal tract. Those in which the infecting organism has multiplied to a large extent in the foodstuff before ingestion will have a shorter incubation period than those in which growth within the intestine has to occur before symptoms are experienced. Preformer toxin in food is likely to act on the stomach and cause rapid onset vomiting. Knowledge of the clinical details of the illness and the presentation of symptoms provide vital clues as to the likely food poisoning organism. It is rarely practicable or necessary to culture for all pathogens in all samples. Relevant tests will be selected in the light of available clinical and epidemiological information. For example, a pathogen may already have been isolated from human specimens examined in parallel with the food. With this, I conclude today's topic. Hope you have understood. Thank you.